Welcome back, and this is our second video on amino acids and proteins. And in this video, we're going to concentrate on protein structure. So this is going to concentrate on the types of bonding that give rise to the specific structures of proteins and ultimately their function. With each of these, you need to be able to describe the bonding interactions that are occurring at each level of structure and what this means for the protein overall. So the first level of structure that we look at with proteins is its primary structure. So the primary structure is the specific sequence of amino acids that make up the polypeptide chain from which the protein is formed. This is essentially the product of the condensation reactions between the amino acid monomers. This structure is stabilized by or comes about from the covalent bonds born between the C double bond OH carboxyl of one amino acid and the NH2 of the next. So remember that this is where we have the new NH C double bond O, this new bond being formed here. Okay, so we have the new amide functional group between two amino acids forming the new peptide bond. So the primary structure itself is determined by the peptide bonds, the covalent bonds, maintaining the specific sequence of amino acids. Okay, so we get the amide functional group. The peptide bond actually refers to the bond between the carbon and the nitrogen that is newly formed during the condensation reaction. As we mentioned in class the other day, by convention, amino acid sequences are written from N to C when we read from left to right. That is, that the final amino acid on the chain will have the amino group exposed. We will have all the amino acids in the sequence, and then the last one that forms the other end of the amino acid chain or polypeptide chain will have the carboxyl exposed. So we have the amino end all the way through the biopolymer that is formed to the carboxyl end. And when we read it, we would give the sequence such as this one, glycine, proline, threonine, glycine, glutamine, serine, lysine, cysteine. We would read it this way and then write it left to right. The primary structure is this specific sequence. If we change the position of any of the amino acids, we have a different peptide. Therefore, we will have a different protein. So the specific sequence from left to right gives rise to our primary structure, like beads on a string. The secondary structure of a protein is where we see folding and twisting of that single peptide chain. Okay, so it is a single polypeptide chain. Okay, so these are essentially that chain twisting and folding to form these secondary structures. This occurs and is stabilized due to the hydrogen bonds that form between the carboxyl and the NH groups of different amino acid monomers within the chain. Okay, so different amino acids within the chain, this is where we're seeing the hydrogen bonds form. There are two structures that we need to be aware of for the secondary structure. This is the alpha helix, this corkscrew-like structure that we see here, and the beta pleated sheet. These occur in different sections throughout the polypeptide chain, so there can be regions of alpha helices and regions of beta pleated sheets or beta pleated strands within the one protein. Okay, these start to give rise to these three-dimensional structures that will determine the overall shape of the protein. The alpha helices form the coils, the beta pleated sheets are the folds. We have the amino acid subunits here where you can see that the NH of one amino acid is forming a hydrogen bond to the C double bond O of another amino acid. These are, however, in the same amino chain, so there would be a coil or a turn in the chain in order for these two to line up to form a beta pleated sheet. With the alpha helices, you can see here that we have the NH of one amide link forming a hydrogen bond to a different peptide further along. This causes the chain to coil in order for these hydrogen bonds to have the right orientation and correct distance. So with secondary structure, the key thing is that we can explain that it comes as a result of hydrogen bonding, and that hydrogen bonding is always between the NH of one amino acid or one peptide bond in the chain to the C double bond O of another peptide group further along in the chain. Once we have these secondary structures, okay, the alpha helix, which is the coiled configuration, and the beta pleated sheet, how these come to fold together. So you can see here that we have a beta pleated sheet with this is just a sequence of amino acids that is a turn, and then this sequence may fold into an alpha helix and so forth like this. These are occurred by bonding along the peptide backbone. Okay, so this here is our peptide backbone. This is where we get the amide bonds forming, and then the hydrogen bondings between those to give rise to our secondary structure. The combination of these different alpha helices and beta pleated sheets then gives rise to our tertiary structure. So we know that primary structure is the formation of the initial sequence, which is covalent bonding. Secondary structure is the alpha helix and beta pleated sheet, which is hydrogen bonding with the amide groups. Then tertiary structure is how these pleats and helices fold together to give the overall three-dimensional structure. So if we're defining the tertiary structure, it is the overall three-dimensional structure of the polypeptide due to interactions between the amino acid side chains, so variable R groups. Remember, we have 20 different amino acids. They could be broken down into whether or not they were ionic, hydrophobic, hydrophilic, hydroxylic, i.e. containing a hydroxyl group, basic, all those different categories. Because we have all those different side chains, we see a large number of different interactions that can occur to stabilize the polypeptide chain. If our groups are aliphatic or hydrophobic, the interactions here are going to be dispersion forces. We will tend to find these on the inner surface of our globular protein. So within a fold, on the internal surfaces, we will tend to find hydrophobic groups and dispersion forces between them. Hydrophilic groups, however, so if they're charged or form hydrogen bonds, such as our serine groups, these will tend to be on the outside of the polypeptide, so on the outside of the protein, where they interact with the aqueous environment of the cell. Dipole-dipole interactions for amino acids that contain sulfur, such as methionine, or our non-protonatable nitrogen groups such as histidine. These will form dipole-dipole interactions, polar groups that do not form hydrogen bonds. The other ones that we have are hydrogen bonds that occur between the side chains that contain OH, NH, or a C double bond O. Okay, remembering that we can have the NH forming a hydrogen bond to the C double bond O, a OH forming a hydrogen bond to a carboxyl as well. So any of these groups which can form hydrogen bonds, same rules as we've seen consistently for all intermolecular forces. The other ones that we tend to see are ionic bonds. These occur when our side chain groups are protonated. So when we see the NH3+, or so the amine group has acted as a base, or where we've seen the carboxyl group acting as an acid, forming the carboxylate anion with the minus sign. These can now have ionic interactions. These are a formal actual charge. Okay, so they are positive and negative. So we see strong ionic bonds, which means that we can also see ion dipole bonds when these interact with polar groups elsewhere in the polypeptide. The last one is covalent bonds. We actually can see covalent crosslinks forming within a polypeptide, and this occurs only when we have two cysteine residues. The two SH groups will actually form a stable disulfide bond or a disulfide bridge, which is what we see here. This helps to stabilize and strengthen the overall three-dimensional shape. So the alpha helices, beta pleated sheets are our secondary structure. The way those subunits fold in to give an overall three-dimensional shape stabilized by the side chain groups is our protein structure. The sequence of amino acids will give rise to a very specific structure. Okay, so the three-dimensional shape of a protein is always specific and comes about due to the interactions from the specific sequence of amino acids from its primary structure. Once we have those, everything we've spoken about so far has been the folding and twisting of a single chain. So the primary structure with our sequence of amino acids, the folding of this into beta pleated sheets and regions of alpha helices, then the overall three-dimensional arrangement of these are all one polypeptide chain. Okay, so they're one strand. Primary, secondary, and tertiary. With quaternary structure, what we are seeing is the interaction between more than one polypeptide chain. So we have subunits which are made up of this peptide, a different peptide, and we can see them in dimers, trimers, or these 4 units as well. So quaternary structure is where we have side chain interactions between multiple tertiary structures. Okay, so we have, again, it's the side chains on the external regions of these folded polypeptides. And then we have multiple subunits coming together to form one single active protein. There's a number of proteins that are examples of this within our body. Collagen, insulin, hemoglobin, all of these have multiple domains, each domain being a single polypeptide subunit. They come together and they're only active when you have each subunit working together to form the overall active structure. So in summary, we need to be able to explain the different levels of protein organization, that being our primary structure, which is the sequence of amino acids held together by covalent bonds in the new peptide links formed from the condensation reaction. Secondary structures, which is the organization into alpha helices and beta pleated sheets stabilized by hydrogen bonds. Tertiary structure, which is the attractions between side chains folding the protein up into an overall three-dimensional shape, giving it its active form. We can also see quaternary structure where we have multiple polypeptide chains coming together to form a larger, more complex unit. So for primary, secondary, and tertiary, we are always talking about within the same chain. Quaternary, we're talking about multiple chains. Let's look at a couple of example questions. This is a multiple choice question. It gives us some information about a protein. It states that jellyfish venom is a mixture of proteins for which there is no antivenom. Jellyfish stings are painful and can leave scars, and in some circumstances can cause death. This is just background. Key thing here is it's telling us we're talking about proteins. Some commercially available remedies disrupt ionic interactions between the side chains on amino acid residues. These products most likely disrupt the proteins and then ask us to pick which structure. Ionic should already tell us that we're interacting with side chains, but it does confirm it further on in the question. This means interactions between the side chains affect only our tertiary structure or our quaternary structure. Primary structure is covalent, so it's not this. Secondary structure is hydrogen bonding, so it cannot be B. D is all three, so it cannot be this. None of our options give quaternary, so it must be tertiary structure only. So looking at a longer response question, let's look at this one from 2014. This one is looking at amino acids, and we're told it can be classified according to the nature of their side chains, in this case called Z groups. You will see side chains interchangeably referred to as R groups or Z groups. Okay, it tells us they can be polar, non-polar, acidic, or basic. We're told to refer to the data book, so you should open up straight away to page 14 and look at our amino acids. And name one amino acid that has a non-polar side chain and one that has an acidic side chain. Straight away, you can look at any of the ones that contain only CH2, CH3 groups. So this could be valine or leucine, okay, anything like that. An acidic side chain, you're going to be talking glutamic acid. Handy that it tells us it's an acid in the name, or aspartic acid would work here as well. Okay, so we've been given amino acids and then the structure of the side chain at pH 7. It's important to look at these and pay attention. We have this one here, the aspartic acid has lost its proton, so it's a negative ion, and the lysine has been protonated. Remembering hydroxyls won't protonate, and the asparagine is not protonated at pH 7. Okay, moving on to the next question, it tells us that the tertiary structure of proteins is a result of the bonding interactions between side chains, just reminding us of that. Use the information provided in the table, where it showed us the different side chains, to identify an amino acid that is involved in the formation of disulfide bonds. This is easy. This one you should always remember. It is going to be cysteine. If we have a look here, it is listed here as having the CHSH group. So cysteine will be our amino acid that forms disulfide bonds, sulfur bridges, always 100% of the time. Two amino acids that may form hydrogen bonds between each other. So we're going to be looking for anything that has NH or OH groups. So let's go have a look. They have given us asparagine, which has a C double bond O NH2. So we could have this one. This is protonated now, so it's going to form ionic bonds, not hydrogen bonds. And then we've got serine. So here we could see we've got the NH2 and the OH. So we're going to say asparagine and serine. Always make sure that you double check your spelling. Okay, so we've got GINE, because VCAR, if they have given you the spelling in the data booklet or it's a chemical name, they expect you to spell it correctly. So always make sure that you check for the spelling on those. Give an example of amino acid side chains that may form ionic bonds, salt bridges between each other. So we're going to be looking for ones that are charged. And we know they gave us some of those because we pointed them out before. So we're going to look for our aspartic acid because it's ionized, and lysine. So these will be aspartic acid and lysine. Identify the type of bonding that exists between side chains of two alanine residues. We go check. Alanine has a side chain of CH3, which means it is non-polar, so this will be dispersion forces only. Okay, the last bit of this question says the enzyme trypsin catalyzes the breaking of peptide bonds. Trypsin is active in the upper part of the small intestine where the pH is between 7.5 and 8.5. Trypsin is not effective in the stomach where the pH is 4. This we will talk about in our next video with enzymes, but essentially what we're talking about here is if we change the pH, we are going to change the environment that these side chains are in. So they may not be charged the same way or have the same interactions. When we do that, we disrupt the interactions between the side chains, which destabilizes the shape, and therefore our proteins don't work as effectively. This is the answers. You have these printed in your PowerPoint, so have a look, and always make sure that you carefully look through the justifications so that you're following what they're looking for in an exam question. Okay, that's it. That's all for protein structure and for amino acids. The next one we'll look at is the activity of enzymes, their role in our body, and how we can alter the effect of enzymes and what causes them to lose function. I'll see you in class.